Nanoworld. The nanoworld refers to the study of and manipulation of structures and phenomena at the nanometer scale, typically ranging from 1 to 100 nanometers. This is a scale so small that it deals with individual atoms, molecules, and the interactions between them. The nanoworld focuses on dimensions at the nanoscale, where unique physical, chemical, and biological properties emerge. It is a domain where quantum mechanics plays a significant role in determining material properties, unlike the classical physics that governs the macro world. Nanoworld refers to a company or concept associated with advanced nanotechnology, specifically in producing tips for scanning probe microscopy or SPM and atomic force microscopy or AFM. These technologies are used in scientific research and industry to examine surfaces at the atomic or molecular level. Scanning probe microscopy or SPM is a branch of microscopy that forms images of surfaces using a physical probe that scans the specimen. It founded in 1981 with the invention of the scanning tunneling microscope, an instrument for imaging surfaces at the atomic level. Scanning probe microscopy refers to a family of microscopy techniques used to examine surfaces at the nanoscale. It utilizes a physical probe that scans the surface of a specimen, gathering information about its topography, mechanical properties, and other surface characteristics. Scanning probe microscopy is a powerful branch of microscopy that creates a high-resolution images of surfaces by scanning them with a physical probe. Unlike traditional optical microscopes that rely on light, SPM uses the interaction between the probe and the surface to produce detailed images at the nanometer or even atomic scale. Atomic force microscopy is a type of scanning probe microscopy which demonstrated resolution on the order of fraction of a nanometer, more than 1,000 times better than the optical diffraction limit. The information is gathered by feeling or touching the surface with a mechanical probe. Atomic force microscopy is a type of scanning probe microscopy that provides detailed three-dimensional images of sample's surface at the nanoscale. It achieves this by measuring the forces between a sharp probe and the sample surface. SPM and AFM tips. These are the ultra-precise tools made to probe and manipulate surfaces. They allow scientists to visualize structures, measure forces, and manipulate matter on a nanometer scale, critical in material science, biology, and nanotechnology.
Okay, so let's move on to the origin of nanoscience. So Richard Feynman was a brilliant physicist who inspired the field of nanoscience. In his talk, There's Plenty of Room at the Bottom, he proposed the idea that we could manipulate and control individual atoms and molecules. But back then, there's no tools existed to make this happen. But his idea was revolutionary. It challenged scientists to think beyond the limits of technology at that time. He was basically saying, if we figure out how to do this, we could create amazing things. And with that word, na-challenge yun ang mga scientists to explore and dig deeper. Kaingon pa ni Feynman, if we could control these tiny blocks directly, we could create new materials like kanang machines or kanang tools that are extremely small but very powerful. So his vision essentially sparked the birth of nanoscience. But kanang take note ha, during his time, wala pa gyud ni exist ang term na nanoscience. Naga-base ra gyud sila sa talk ni Feynman, dili pa gyud diri formally gasugod, but his foresight motivated scientists and engineers to think about how tiny particles could have a huge impact on technology. And then just a year after Feynman's talk, engineer Mohammed Atala and Dawon Kang made a breakthrough that brought nanoscale work into reality. So bali ari na naghinay-hinay og kanang sugod og buhat og mga products about ana. At the Bell Labs, they invented the MOSFET, a small yet powerful transistor used to control electrical signals in devices like computers. So the MOSFET's design included components as small as 100 nanometers thick. So para mas masabtan or mas ma-envision ninyo kung unsa na siya ka bag-on, when we say nanometer, it is 1 billionth of a meter. Meaning, very nipis kaayo ni siya, very gamay, para mas ma-imagine ninyo. Kung i-compare na to siya sa usa ka strand sa atong buhok, mas kuan jud kay siya, mas nipis o gamay to the point nga dili siya makita through our naked eye lang. So balik ta sa MOSFET. So MOSFET is one of the early example of a nanoscale engineering. Their work was crucial because the MOSFET became the backbone of modern electronics, including computers and smartphones. So it also showed how nanoscience could be applied to solve real-world problems and making smaller and more powerful devices. Next is nanotechnology. So in 1974, a scientist named Norio Taniguchi came up with the word nanotechnology. He used this term to describe working with materials and devices that are very, very small. Like pariha sa akong gingon ganina, it is much smaller than what we can see with our eyes because it is a billion times smaller than a meter. So his work focused on manufacturing very thin layers of materials, like in electronics, where these tiny layers are important for creating devices like computer chips. So at this point, he was the first to understand how important it could be to work with such small parts and make them with great precision. So even though Taniguchi wasn't talking about the high-tech devices we have today, his ideas of working with tiny things was the start of nanotechnology. So, and then in 1986, K. Eric Drexler, an American scientist, expanded the idea of nanotechnology. He imagined a future where we could build tiny machines that could work at the atomic level. He thought of nanoscience assemblers. It is a machines that could create anything by putting atoms together, and just like building Lego blocks ingon ana. So for example, these assemblers daw kuno could build new materials like repair human tissues or even clean up pollution. So Drexler's ideas made people excited about how nanotechnology could transform fields like medicine, technology, and the environment. His ideas were more futuristic, showing how nanotechnology could do much more than just creating small things. Drexler helped people think of nanotechnology as a powerful tool that could solve big problems and make significant changes in many areas of life. Take note ha, theory ra ni Drexler. Dil ka nang he was just imagining kung unsay potential na mahitabo sa future if we really dig deeper, like i-explore yun natin pa maayo ang nanotechnology. But even though he wasn't physically building these technologies himself pa, his visionary thinking played a big role in shaping how we view nanotechnology today. Okay, next is nanoscience and nanotechnology. So nanoscience and nanotechnology are fields of science that deals with studying and creating things at an incredibly small scale called the nanometer scale, which is a billionth of a meter. Pareha sa kung gingon, it is much smaller than what we can see with our eyes. So, nanoscience focuses on understanding how tiny materials such as atoms and molecules behave and interact when they are so small. So, these tiny materials often have special properties that we don't see in bigger materials. Nanotechnology, on the other hand, takes the knowledge gained from nanoscience and uses it to make new products or solve problems. Like, for example, nanotechnology helps create tiny medical devices that can target specific areas of the body to treat diseases, or special coatings that make things like clothes or electronics resistant to water or dirt. So this technology also makes things like computer chips smaller and more powerful, so allowing for fast and better devices. So in short, nanoscience is about studying how tiny things work, and then nanotechnology is about using that knowledge to make new things that are helpful in everyday life. So ari sa nanotechnology, ari nag-work, naghimo nag product para kanang maka-help sa atong everyday life, like environment baron or para sa atong kaugalingon, like for medicine ingon ana, like na-apply na nimo diri ang imong nakatunan.
Nanotechnology is the study and use of things that are super tiny, as small as just a few atoms. So it happens at a size called the nanoscale, which is about 1 to 100 nanometers. To imagine how small this is, think of a human hair. It's about 80,000 nanometers wide. Scientists use nanotechnology in different areas like medicine, electronics, and materials to create new and useful things. So, we have here an example. Nanotechnology is used in making lightweight but strong materials for sports equipment or even in sunscreens that spread better on your skin. Now we have here nanometer. A nanometer is a unit to measure very, very tiny things. It is 1 billionth of a meter. If you think how small an ant is, a nanometer is million of times smaller. So it's small. We need special tools like microscopes to see anything at this size. So for example, DNA, the molecule that carries your genetic information, is about 2.5 nanometers wide. Next, the nanoscale is the size range where nanotechnology works from 1 to 100 nanometers. At this size, materials start behaving in surprising ways, like becoming stronger or changing color. So because the rules of physics and chemistry are different at this tiny level. So we have here an example. Gold looks shiny and yellow when beaten, but at the nanoscale, it can appear red or purple and is used in medical tests. Next, we have here the nanostructure, are the tiny shapes or system created at the nanoscale. They can be nanoparticles, nanotubes, or nanowires, depending on their shape. So these are designed to do specific jobs, like delivering medicine only to sick cells or making computer chips faster. So for example, the nanostructures are used in drug delivery systems to target cancer cells without harming healthy ones.
So let's move on the aspects of gene therapy. So gene therapy is a potential method to treat or cure genetic diseases that cannot be cured through traditional medicine. So researchers were able to restore basic hearing in deaf mice using gene therapy by introducing a gene through the cochlea. So gene therapy is a way to treat disease caused by problems with our genes. So it's like fixing a mistake in our body's instructions. Regular medicine can't always fix this genetic problem. So example, scientists were able to help deaf mice hear again using gene therapy. So they added a gene to the mice's inner ears cochlea and they fixed the problem that was causing their deafness. For our next topic is about the aspects of gene therapy. So what are the aspects of gene therapy? So gene therapy is like a powerful tool that can help cure diseases that traditional medicine can't fix. It's a new and exciting field and it's already helping people with some diseases. So gene therapy tries to fix these broken genes in 3 ways. So the first one is gene replacement, that's swapping a broken toy part for a new one. So we will replace the faulty gene with a healthy copy. And the second one is gene inactivation. So this is like turning off a broken toy part that's causing problems. So we disable the faulty gene so it doesn't cause any harm. So the third one is gene addition. So this is like adding a new toy part to make it better. We add a healthy gene to help the body work correctly. At the next page, take a look at the picture. So this image shows different ways that gene therapy works. So the basic idea is to correct faulty genes that cause diseases. So the one method, the top left, shows replacing a faulty gene inside a cell's nucleus with a healthy one, so leading to the production of a normal protein instead of a defective one. And another method, the bottom and the label ex vivo gene therapy, shows how cells are removed from a patient's body, modified in a lab to have a healthy gene, and then returned to the patient where they produce the needed proteins. So a similar process is showing using bone marrow. So the image on the right shows a gene being inserted into a cell, so correcting its function. So in simple terms, gene therapy aims to fix the genetic instruction with then cells to cure or treat diseases. And the next topic is gene therapy. So medical science has detected many human diseases related to defective genes. So these types of disease are not curable by traditional methods like taking readily available medicines. Gene therapy is a potential method to either treat or cure genetic related human illness. Many diseases are caused by defects in our genes. So these genetic diseases often can't be cured by regular medicines like pills or injections. So gene therapy is a new approach that tries to fix these gene defects, offering a potential cure or treatment. For example, some genetic diseases affect how our bodies make proteins, leading to problems with different parts of the body. So gene therapy aims to correct the gene instructions so the body can make the correct proteins and function normally. Let's move on the basic process. So the basic process, there are several approaches to gene therapy. So these are the following: Fleischer at 2017. So replacement of mutated gene that is cause disease with a healthy copy of the gene. So inactivation of a mutated gene that is functioning improperly. So introducing a new gene into the body that fight a disease. So here's a simple explanation of the basic process of gene therapy. So gene therapy tries to fix problems caused by faulty genes. So there are the few ways to do this. So first is replacing a bad gene. So a healthy copy of the gene is used to replace the one that's causing the disease. And the second one is unactivating a bad gene. So if a bad gene isn't working correctly, gene therapy can be used to turn it off so it doesn't cause any problems. And the last one is adding a new gene. A new gene can be introduced to help the body to fight diseases.
Two types of gene therapy. The first one is the somatic gene therapy. Involves the manipulation of genes in any cells of the body except sperm and egg cells that will be helpful to the patient but do not inherited to the patient's descendants. So this type of therapy works on the non-reproductive cells of the body like skin cells, blood cells, or muscle cells. Scientists change or replace faulty genes in these cells to treat or prevent diseases. The change made during this therapy help the patient but do not get passed down to their children or future generations. For example, if a person has a genetic disorder affecting their immune cells, somatic gene therapy can target and fix those immune cells to make the patient healthier. Well, the second one is germline gene therapy. Involves the genetic modification of reproductive cells such as sperm and egg cells that will pass the change onto the next generation. So this therapy focuses on reproductive cells like sperm or egg cells or an early embryos. So the goal is to correct or modify faulty genes so that the changes are passed into future generations. So this means is that the offspring of the treated individual will inherit the corrected gene. Example, if a genetic mutation causes a hereditary disease, fixing the genes in the sperm or egg cells can prevent the disease in future children. Somatic gene therapy affects only the treated individual, while the germline gene therapy affects not just the individual but also their descendants. So that is the two types of gene therapy. And before we proceed to the next reporter, I have questions. Have you heard stem cells in the news? If yes, perhaps you wonder if they might help you or your loved one with a serious disease. You may wonder how they're being used to treat diseases and why they are being discussed so vigorously. Stem cells are mother cells that have the potential to become any type of cells in the body, under the right conditions in the body or a laboratory. Stem cells divide to self-renew or multiply. They can become cells of the blood, heart, bones, skins, muscles, brain, and among others.
The next topic discusses stem cell gene therapy. Stem cell gene therapy focusing on the potential of stem cells as mother cells that can develop into various cell types in the body. Stem cells possess the unique ability to divide and self-renew under the right conditions, whether in the body or laboratory setting. This property allows them to generate specialized cells, including those of the blood, heart, bones, skin, muscles, and brain. Stem cell gene therapy leverages this characteristic to treat diseases by replacing damages or regenerating tissues. This emerging medical approach holds great promise for addressing conditions like genetic disorders, organ damage, and degenerative diseases. The next slide describes the sources of stem cells, dividing them into two main categories: embryonic stem cells and somatic stem cells, or what we call somatic adult stem cells. Embryonic stem cells are harvested from embryos in the blastocyst phase, typically from surplus embryos created during vitro fertilization, or what we call IVF procedures. These cells are pluripotent, meaning they can differentiate into almost any cell type in the body. Somatic stem cells, on the other hand, exist throughout the body after embryonic development and help maintain and repair tissues. These cells can or have the ability to renew themselves and can differentiate into specific cells type. These cells have the ability to renew themselves and can differentiate into specific cell types related to the organ or tissue from which they originate. And that would be all. Thank you for listening.